Thanks very much. It's great to be here again. This is my second SLC6A1 Connect meeting. And I am going to talk about, there's too many people to thank, but I think we'll go, as I go along, hopefully I can do that. I want to talk about a potential therapeutic strategy that's based on novel allosteric activators of GABA transport. And I'll explain what those are for those of you who aren't familiar as we go along. But I want to state at the outset that this work really depended on this meeting last year, in the sense that meeting with people and talking to people like Caddy and Dr. Demarest, Dr. Greenspan, I got some ideas and went back to the lab to test some ideas. I'll explain as we go along. This slide, you might remember, I think I showed it last year. Basically, it shows how we can study GABA transporters in an exogenous cell expression system that is very useful, the Xenopus oocyte system. You can inject RNA into the oocyte, and you can inject wild-type GABA transporter RNA together or alone with mutant GABA transporter RNAs. And when we started looking at the effect of the S295L mutation, we found that the mutation itself had a profound effect on the GABA transporter, on SLC6A1. It abolished uptake. But perhaps more interestingly, we found that when it was co-expressed with SLC6A1, when the mutant transporter was co-expressed, it had no dominant negative effect on uptake by the wild-type transporter. And the reason this is important is because it kind of directs some validation towards the concept of increasing SLC6A1 expression in the context of a loss-of-function disease, like using ASOs, for example, or viral constructs. We also use the transgenic mouse model using a knock-in mutation that mimics the human mutation S295L. And when we started to look at kind of some of the very basic biochemical effects of the mutation, and we used really more or less crude assays, but assays that aren't used very commonly any longer. They used to be 30 years ago. And maybe I'm kind of dating myself. But we found that the wild type transporter brain slices acute sections that we keep alive in artificial cerebral spinal fluid. They would mediate this amount of uptake, compared to what you can see in the next column is uptake mediated by the heterozygous mouse with one copy of the S295 allele. And then the homozygous mouse with two copies of the mutant allele. And then you can see on the far right the effect of the SLC6A1 selective antagonist SKF89976A. And what that shows is that blocking all of GABA uptake with all of SLC6A1 mediated GABA uptake gives us the same level of total uptake as is seen with the homozygous mouse. And the heterozygous mouse, which really is a model for the patient, the amount of uptake is intermediate between those two levels. And also notable is the fact that really the majority of GABA uptake is not lost, even losing all SLC6A1 mediated GABA uptake. And we'll talk a little bit later about what mediates the rest of that uptake, but a part of it is SLC6A11, GAT3, and then there's another transporter that we haven't molecularly identified yet. And the evidence for those three components is here. You can see that in control conditions, now this is in a wild-type mouse brain tissue, the amount of uptake seen when you block all SLC6A11, or SLC6A1 rather, with SKF is shown here. It's reduced about 25%. If we further block with SNAP, which blocks SLC6A3, or A11, which is GAT3, sorry about the nomenclatures, it's blocked down further, about 60% block. And then if we use a high concentration of a non-selective GABA transporter blocker, we can block the rest of it. So it's what's blocked by nipicotic acid at high concentrations can also be blocked by high concentrations of unlabeled GABA. So we're calling that low affinity component GAT-X. We don't know what it is. We're actively trying to molecularly identify it. But it's interesting that 30 years ago, a very similar report was published in J Neurochem by Wood's group that reported these three components, and the affinities of each component match well with GAT1, GAT3, and GAT-X, as we call it. So I'm going to talk really briefly. I'll skip over this slide, but I just wanted to show you've seen data presented by other groups now that mice that are that express loss of function SLC6A1 genes have seizure activity. They also have these spontaneous spike discharges. And this is an example of recordings that we do in not in vivo, but in vitro brain slice preparation, using recording electrodes to record extracellularly. We can see a relatively modest increase in seizure activities in the heterozygous mice, and a much more profound and obvious change in these spontaneous spike discharges in homozygous mice. So based on this work and work you've heard about already, it's not a stretch to imagine and theorize that loss of function SLC6A1 mutations might result in increased levels of extracellular GABA in the brain. And we set out to test that by measuring tonic GABA A receptor activity. And Kirill talked a little bit about how this is done earlier. I won't go through the details, but I want to just summarize the findings. What we found in this case, recording from entorhinal cortex, we find evidence that the amount of tonic or ambient GABA A receptor activity is increased relatively strongly in the homozygous mutant animals, and it's increased to an intermediate level in the heterozygous animals. And based on the kinetics that we know about the GABA A receptors, we can estimate that the increase in concentration of GABA in the extracellular space caused by the heterozygous mutation is roughly doubling the concentration of GABA, because the level of activity of the GABA A receptors is approximately doubled. So there is it sounds like a lot, but the concentration of GABA normally is really quite low in the brain, so this is a doubling of a relatively low concentration to start with. And also, Kirill talked a little bit about synaptic responses, synaptic transmission in the transgenic mice. And what we did was look at miniature spontaneous inhibitory postsynaptic currents that are mediated by GABA vesicular fusion that occurs spontaneously, and we can record in postsynaptic neurons using a whole cell patch clamp technique. And if we compare the wild type heterozygous and homozygous iPSCs, we find that they're not significantly altered by the mutation. And I'm not going to show the data, but our data agree with Kirill in looking at other types of iPSCs that reflect ensemble release of GABA from multiple terminals. If one can look at spontaneous iPSCs or evoked iPSCs, and those are larger in the mutant animals. But I wanted to show this because I think, and highlight it because I think it's important, because what it shows is that the vesicular machinery that underlies GABA synaptic transmission is essentially altered in the mutant animals. And this is interesting and important because SLC6A1 has always been kind of referred to as a transporter that's on presynaptic GABA nerve terminals. And it was thought to be critically involved in reuptake of transmitter, recycling of transmitter, to allow repackaging in vesicles and to allow continued synaptic transmission. But this data shows that there are clearly other mechanisms that neurons use to recycle GABA and refill synaptic vesicles of GABA. And it's good from the standpoint of trying to find patient treatments. It means that there's kind of a fundamentally underlying, the molecular machinery for GABA synaptic transmission is unaffected. We've looked at a number of other types of neurophysiological changes in the mutant animals, and I'm not going to go through all of them. I'm just going to show you one or two. This is an important neurophysiological assay. You've probably seen it. It's Schaefer collateral CA1 long-term potentiation, which can be studied in the hippocampus. If we record EPSCs in postsynaptic neurons in CA1 region of hippocampus, we stimulate presynaptic axon to release glutamate. And if one gives a brief high-frequency stimulus, you can see that the level of synaptic transmission goes up to a level that is increased and is maintained for as long as you can keep a slice alive in a dish. And this is basically the kind of well-accepted cellular molecular model for synaptic plasticity, learning, and memory. And so what we see is that there is a reduction in LTP in the both heterozygous and homozygous SLC1, S295 mutant animals. So we've been hearing a lot about the work going on in this Ravicti trial. You've heard from Dr. Greenspan, and Dr. Demers is here as well. And the data, and even the anecdotal data that's not published yet, is very exciting. And after speaking with both of them last year, I came away really interested in also reading Caddy's paper on phenylbutyrate and its actions on rescuing GABA transporter mutant phenotypes. I was stimulated to kind of try to look at this in more detail. Actually, it was Scott who we were sitting down after lunch, and he said, after my talk, he said, it looks like you kind of know what you're doing. You should try to figure out what phenylbutyrate is doing. And I said, yeah. I mean, we went at it and expected that it was going to be. It's obviously known to be a chemical chaperone, and we have no evidence that it's not a chemical chaperone. But in our hands, we didn't find evidence that phenylbutyrate increased GABA transporter expression, at least in the model that we looked at, which is different than the cell models that have been published. But what we found was kind of unexpected and interesting. We found that when phenylbutyrate is co-applied with GABA, that there is an increase in GABA transport activity. And if you notice the time scale, that's 50 seconds, that bar. The increase in activity occurs rapidly and it washes out very rapidly. And this is inconsistent with a mechanism that would involve a trafficking change of the protein or any kind of metabolic or biochemical change. And this increase in current that we see in the xenopistoocyte is also tracked by measurements of radio-labeled GABA uptake that we make in the cells in the presence and absence of phenylbutyrate. So we were very interested in this. There hasn't really been, there are a number of allosteric activators for other targets. And, probably the most well-known one is a very common drug. The whole class of benzodiazepines are actually allosteric activators of GABA-A receptors. That's how they work. Allosteric activators bind at a site on a target protein, receptor or channel, in this case a transporter, and they either increase or decrease its efficacy. We decided to start looking immediately at other analogs of phenylbutyrate, and I'm not going to show you all these data, but just as an example, we found that if you change the chain length of the butyric acid side chain on the phenylbutyrate, that this allosteric activation was preserved. And we've gone through and tested a whole a range of analogs of phenylbutyrate and found varying degrees of efficacy and affinity. So this is kind of cool from the standpoint of drug discovery because it gives us a platform to modify and test, and even in a small lab like mine, we can do semi-high throughput screening. I want to show you maybe the most interesting thing that I think we've found so far, which is we asked what happens if you just get rid of the phenyl group and look at butyric acid alone. And butyric acid by itself actually works as well, actually a little bit better than phenylbutyrate at its actions as a positive allosteric modulator of GAP1, SLC6A1. You can see that the currents are increased, and the increase is proportional to the increase in radiolabeled GABA uptake in the cells. So yeah, we can do these sorts of systematic drug discovery efforts, varying the structures of these molecules, and try to figure out what the active pharmacophore is. And these are experiments that are still underway. We also, of course, wanted to test these molecules at SLC6A11 or GAT3, and we found that interestingly, they had actually larger effects on SLC6A11. The increase in GABA uptake mediated by GAT3 was increased, as you can see, to levels approaching threefold, whereas in GAT1 it was more on the order of 1.5-fold. So these are experiments in vitro in cells expressing the transporter. Now, what we wanted to, of course, do is see what these would do in the brain, and we looked at GABA uptake in brain tissue from these experiments show uptake in brain tissue from wild type and heterozygous SLC6A1 S295L animals. And what's shown here is just kind of a really abbreviated dose response with 4-phenylbutyrate on the left and butyrate on the right. And you can see that compared to the level of uptake in the wild type brain section, if we use low millimolar concentrations of butyric acid, 0.3 or 1 millimolar, we can restore the normal level of GABA uptake in the brain slice of the heterozygous animal. So this gives us a lot of encouragement to pursue looking at this as a potential therapeutic molecule, because butyrate itself is a naturally occurring short-chain fatty acid. It's actually produced in our own guts at low levels. It's produced by the microbiome. It's a constituent in butter and cheese. And of course, it doesn't cost $800,000 a year. We're also in the middle right now of looking at butyrate's actions on restoring some of the neurophysiological deficits that we see in the mutant mice. And this just shows an example of an experiment I didn't talk about yet, but what we're looking at is the excitability of neurons. These are neurons in entorhinal cortex again, and we're injecting small amounts of current to elicit trains of action potentials in the neurons. And this is a common technique to basically measure excitability or excitation spike coupling. And we can see that in the heterozygous and even more in the homozygous mutants, there's a deficit in this excitation spiking coupling. And that's plotted in the middle. And what we see it's exciting is that if we look at the heterozygous neurons, or neurons from heterozygous animals, butyrate returns, it left shifts this excitation spiking coupling curve to levels that are the same as in the wild type mouse. So we are, in the middle now of doing many other experiments to look at the neurophysiology of the mutant animals and testing whether butyric acid and some of these other fatty acid analogs can restore normal neurophysiological function. So I'm not going to read all these conclusions. You can read them. I think that I just want to highlight the last three, which are that the chemical chaperone, phenylbutyrate, and other fatty acids and analogs act as direct positive allosteric modulators of the GABA transporters SLC6A1 and SLC6A11. And this was a surprising finding that hadn't been reported before. So we went back and actually found that there was a paper that sort of got buried in the literature. I'm just going to show a slide I wasn't planning to show. I hope I have a minute, 30 seconds. Valproic acid is a butyric acid derivative. It's a pantanoic acid. It's an isopropyl pantanoic acid. And Sepp Eskandari, a colleague, so I'm ashamed to have not really noted his paper a number of years ago, published that valproic acid is an allosteric activator of GAT1, SLC6A1. And we went back, of course, and repeated those experiments, and we found, indeed, that it is an allosteric activator, and we believe it works similarly to the other butyric acids and analogs. It actually is a slightly more efficacious positive allosteric modulator. And, I think, at least to my understanding, anecdotally, valproic acid has had sort of mixed success in treatment of SLC6A1 disease. In some cases, it helped, or might have helped for a short time, and then it stopped. Then, of course, it's important to remember that valproic acid itself, it has many other actions, and those are, off-target actions in the sense that it does other things than act on SLC6A1. So we're heartened to report this, that butyrate at low millimolar levels can restore basically normal levels of GABA uptake, and we hope that by treating animals, and now we're also in the midst of some in vivo experiments with mice to give them butyrate to hopefully restore GABA homeostasis in the SLC6A1 mutant animals and assay their behavioral neurophysiological effects. And I'd like to really talk to a number of you that Gene, for example, I want to talk to you guys about collaborating to look at the effects of these PAMs, these positive allosteric modulators, in different types of assays, including behavioral assays. And then finally, it's interesting, this is the structure of butyrate. And as I said, it's available as an over-the-counter supplement and it's sold as a supplement for gut health. And it has interesting actions on that, and it also has actions as a mitochondrial substrate for energy. And shown next to it is a triglyceride analog of butyrate, which is sort of analogous to Ravicti and phenylbutyrate. And this analog tributyrin is also available over the counter as a supplement. And so both of these compounds offer, I think, interesting starting points to try to to try to continue research into testing their potential application for therapeutic treatments. So finally, I want to thank the folks in my lab that did this work, Allie Jester, who's here, and two other students, Andrew and Patrick, and collaborators at UM, including Greg Leary. And also I wanted to thank Scott and Zach for inspiration, and also because we're beginning just now to start to talk about how we could potentially go about moving towards a trial. And then I also want to thank SLC 6A1 Connect for the support, and Amber and Kim and others in the Patient Advocacy Foundation, and NIH for a grant that I'm sort of stealing money from to do this research. So thank you very much.